is Bre-Shea and this is the tryptophan operon. When the operon is on, RNA polymerase binds to the promoter, which allows the RNA polymerase to flow through the genes of the operon, which is a process called transcription. The tryptophan repressor is transcribed, creating an mRNA segment. The mRNA segment is translated into a protein repressor, which is inactive because tryptophan is absent when the operon is on. When the operon is off, tryptophan binds to an allosteric site on the repressor, causing the repressor to become active. The active repressor binds to the operator, blocking the RNA polymerase from binding to the promoter. Therefore, no RNA can be made. This is the LAC operon. When the LAC operon is off, lactose is absent. LAC-I is transcribed into an mRNA segment. The mRNA segment is then translated into a protein, which is the active repressor. The active repressor then binds to the operator, blocking the RNA polymerase from binding to the promoter, which means that the three enzymes cannot be transcribed. The three enzymes need to be transcribed in order to metabolize lactose, making the operon, when it's off, a waste of energy. When the operon is on, lactose is an inducer and it binds to the active repressor, changing the shape of the active repressor, allowing it to not be able to bind with the operator anymore. RNA polymerase can now transcribe the three enzymes, metabolizing lactose. LAC-Z codes for beta-galactosidase, which hydrolyzes lactose into glucose and galactose. LAC-Y codes for permease, which is a membrane that allows lactose to flow into the cell. LAC-A codes for transacetylcolase, which removes the acetyl group and breaks down lactose. This is the CRP operon. The CRP operon is the same as that LAC operon. It has the LAC-I. There's two things that can go on in the CRP operon. You either can have high levels of glucose and low levels of CAMP, or high levels of CAMP and low levels of glucose. In this case, we have high levels of CAMP. The CAMP binds to an allosteric site on the CRP, changing the shape of the CRP, turning it into an active CRP. The active CRP binds with the RNA polymerase, and the two proteins work together to speed up the rate of transcription of the three enzymes. When there is absence of CRP and high levels of glucose, the process still goes on, just at a much slower rate. RNA splicing begins with pre-mRNA. Pre-mRNA is composed of introns and exons. Exons are expression genes and introns are non-coding segments. We don't need the non-coding segments in creating an mRNA, so the introns have to be eliminated. This is done by SNRPs. SNRPs are composed of sRNA and proteins. Many SNRPs and other proteins are composed together to create spliceosomes. The spliceosomes attach to the ends of splice sites, eliminating the introns, leaving the exons left over.